Hi, my name is Natalie, and I'm here to talk to you about muscle contraction.
Within each muscle are fascicles, groups of muscle fibers. Each muscle fiber is made up of myofibrils, and within the myofibrils are sarcomeres, individual structural units of muscle. Within each sarcomere, there are thick and thin filaments. The thick filaments are made up of myosin proteins, and the thin filaments are made up of actin proteins, troponin complexes, and tropomyosin proteins. Notice the Z line. It will be important later.
For a muscle to contract, a neuron or nerve cell will send an impulse to the muscle
in the form of an action potential at the neuromuscular junction. This causes calcium ion channels in the sarcoplasmic reticulum membranes to open, located between myofibrils within each muscle fiber. When the muscles relax, the troponin complex blocks the myosin binding sites on the actin. The calcium ions diffuse into the myofibrils and bind to the troponin molecules. Once the calcium has bound to the troponin changes shape, causing the tropomyosin to change its position on the actin filament. At this point, the myosin binding site on the actin is now free, and the myosin heads of the myosin filament can bind to the actin. ADP and phosphate attached to the myosin head are released when the myosin head binds to the actin filament. Now, the myosin heads can pull the actin filaments towards the center of the sarcomere, contracting the muscle. The binding of another ATP molecule to each myosin head causes them to let go of the actin. Every myosin head breaks down ATP to ADP and phosphate, releasing energy that extends the head again, and the cycle repeats with each new nerve impulse. Without nerve impulses or calcium, the myosin filament is unable to move the actin filament, so the muscle relaxes.
You might think that muscle shortens during muscle contraction, and while that may be true, the actual filaments within the myofibrils do not shorten. Let's look at a model of a full sarcomere.
Imagine if the same pulling motion was applied on this model. That would cause the filaments to slide past each other and the Z lines on either side of the sarcomere to grow closer to each other. This sarcomere model, made of candy, frosting, and cake, is delicious. Do you know what else is delicious? Animal muscle, which you eat whenever you eat meat.
Let's take a moment to talk about death real quick. When you die, you stop breathing, which means your body no longer takes in the oxygen it needs to make ATP. Without any more ATP, the actin and myosin filaments can't slide away from each other, so your muscles stay contracted. This means the entire body goes stiff, which is called rigor mortis. So in CSI, when the coroner says that rigor mortis has set in, this means that the body has gone stiff, and it is nearly impossible to move limbs along their joints. Rigor mortis ends eventually, however, not because the muscles relax, but because autolysis takes over. Autolysis is a self-digestion of the body cells, causing muscles to break down and soften before further decomposing. Thank you for taking some time to learn about muscle contraction with us. We hope you find it very interesting.